Okay, so good day again. So this is the continuation of our lecture. So a while ago, we discussed about adaptive immunity. Now we will be discussing about the acquired immunity, otherwise known as the non-specific immunity. So let me tell you that these are the different types. Okay, uh, let me have a blank screen. These are the different types of acquired immunity. Okay. Acquired immunity can either be active or passive. Okay. Under the active or passive, it can either be naturally acquired. So passive immunity could also be naturally acquired, or both of them can actually be artificially acquired. Actually, we have already discussed this in bacteriology, but let's recall them, okay? So, naturally acquired immunity happens when you recover from infection. So, when you recover from infection, you'll develop antibodies. And because of an amnestic response, because of the anamnestic response, your memory cells can remember previous encounter with that particular pathogen, and you're now immune to the said type of infection. Okay, so that is the naturally acquired immunity. However, it is not applicable in all kinds of infection. Let's say, for example, COVID-19. So once you are infected with COVID-19, it doesn't necessarily mean that you have already a naturally acquired immunity against your infection. No, that's not true, because there are different variants of COVID-19. Okay, but it can still happen. Okay, so very minimal studies on the natural acquired immunity for COVID-19. Artificially acquired active immunity means that this usually happens as a result of immunization. Okay, so let's say, for example, vaccine. So in vaccine, the antigen is introduced to you, and you train your body, your immune system, to produce antibodies against that particular pathogen. So again, you have several doses and even booster, because again, you are banking on the possibility of an amnestic response because of our memory cells. So the common denominator, therefore, of active immunity is the presence of an amnestic response, and at the same time, it is called active because your immune system, particularly your plasma cells, produce antibodies. In passive immunity, antibodies were just given to you. So, for example, in naturally acquired immunity, you just inherit maternal antibodies when you were born, because as a neonate, you are not yet capable of producing your own antibodies. And majority of these antibodies are IgG, because it is the only antibodies that can pass through the placenta. So IgG is the only antibodies that can pass through the placenta. Okay, so artificially acquired passive immunity, antibodies were just given to you. So what's the difference between artificially acquired and active and artificially acquired passive? In artificially acquired active, you are given an antigen. In artificially acquired passive, you are given antibodies. And these antibodies could be in the form of antitoxin, or in or or serum antibodies, yung tinatawag natin, for example, convalescent plasma, yung mga plasma ng tao na naka-recover from COVID-19. They have a lot, a lot of, antibodies, or anti-rabies is another example. So all of them are actually considered to be as artificially acquired antibodies because antibody are artificially acquired passive. So pag sinabing passive, your immune system does not play an active role in the production of antibodies. So the common denominator of passive immunity is that there's no unamnestic response. So passive immunity is not long lasting. It is just short lived. And at the same time, since it is not long lasting, okay, it is not long lasting. So, but the action is immediate, the result is immediate. Hindi katulad ng active immunity that it takes time before you produce antibodies. Sa passive immunity, nandito na talaga yung antibodies. So, because of that, passive immunity can be therapeutic. Both of them, can be therapeutic. Okay? Unlike artificial active immunity, it is not therapeutic, but more of prophylactic. Etong passive is therapeutic and prophylactic at the same time. Okay, so that's the difference. These are the different types of acquired immunity. Okay, so I hope that's clear. So we're just still in the introduction. Now, the acquired immunity is actually the adaptive immunity wherein we will be dealing with more of cell-mediated immunity. So that's the reason why one of the topics that we will have here is about T cell differentiation. And then we will also be discussing about humoral immunity. And that's the reason why one of the topics here is the B cell differentiation. So the term differentiation, these terms refers to the maturation and development of T or B cells. So when you say differentiation, we are referring to the maturation and development. So that's why they will have different roles in adaptive immune responses. So adaptive immunity is specific to each pathogen or microbial agent. So that's the reason why it is also known as specific immunity. And one of the most important aspects of adaptive immunity is that it can remember prior exposure. So we call it an amnestic response. And it is the main responsibility of the memory cells. Okay? It increases response to a pathogen upon repeated exposure. So increased response is manifested by an increase in antibody titer. So again, when you say an increase in antibody titer, this is synonymous with an increase in antibody concentration. Okay, it takes longer to become activated, particularly when we talk about active immunity. But the most important thing about adaptive immunity is it is longer lasting. And this can involve T cells and B cells. Okay, so T cells are important for cell-mediated immunity, while B cells are important for humoral immunity. Okay, so let's talk about the T cell differentiation. So in the development and maturation of T cell, okay, the first stage is called the double negative stage. Okay, so this is what you call the double negative stage. T cells are both T cells and B cells are produced from the bone marrow. However, T cells will need to migrate to thymus. Okay, thymus is the site where T cells are developed. And the first stage in the development of T cell, we call it the double negative stage. Where in thymus, particularly here at the medulla. Okay? So, this is the cortex, and then this is the medulla. Okay. Thymocytes, in this particular double negative stage, thymocytes undergo rearrangement of genes coding for the TCRs. Okay? Ano ba yung mga TCRs? These are the T cell receptors. So, what do we mean first by thymocytes? Okay. Thymocytes are young T cells, but they are committed to become a young lymphocytes, but they are committed to become T cells. So they are thymocytes. And they need to undergo rearrangement of gene coding for the TCRs. And in the future, there will be two important TCRs. Okay? These TCRs are the CD4 and the CD8. And as we all know, T helper cells would have the CD4, while cytotoxic T cells will have the CD8. Okay, so there is a need to undergo rearrangement of gene so that once the receptors are fully developed, then they are now mature, and mature T cells with fully developed receptors can now exit the thymus. So, just so to be clear, T cells are produced from the bone marrow. However, the thymus is the site of the development of the T cells. Okay. So, at the early stage of the T cell differentiation, we call it the double negative stage. A while ago, I told you that a fully developed T lymphocytes would have either CD4 or CD8. However, during the double negative stage, we will not be able to see immediately the CD4 and CD8. But what we can see here during the double negative stage is called the Cd3 Tcr complex. Okay. Now, as you can see in the illustration, the Cd3 and Tcr complex can actually be consisting of several types of chains. And two of them is called the alpha and the beta chains. Now, if you will be looking at the picture here, it is only the alpha and the beta chains that will have groove. The rest do not have groove. And the reason why they have groove is because they can recognize antigen. So remember, at the early development of the T cell that is called double negative stage, the Cd3 Tcr complex has 6 chains, but it is only the alpha and beta chains that can recognize the antigen. Okay. 6 other chains in 3 pairs that assist with signaling when antigen binds to T cells. So, other chains are important for signal activation. So, how many chains are important for signal activation? 6. So, this is 1, 2, 3, 4, 5, and 6. But only the alpha and beta chains are important for antigen recognition, while the 6 other chains are important for signal activation when antigen binds to T cells. Okay. Why signaling is important? Signaling is important. Number one, why it is important? To alert other cells. And why do we need to alert other cells? So that other cells will be able to produce the essential cytokines needed for the activation, let's say in this case, activation of the T lymphocytes. So there's a signal alerting other cells that, hey, an antigen is recognized. Okay. Eventually, during the double positive stage, thymocytes can now express CD4 and CD8. And they will now rearrange gene coding for the alpha chain. So let's go back to the illustration here. So when the CD4 and CD8 are not yet expressed, we have the CD3 Tcr, CD3 Tcr. It means that it's still in double negative stage. Eventually, thymocytes are exposed to antigen and they're now able to express CD4 and CD8. It is now the double positive stage. Okay. So, double positive stage is an the indicator of having a double positive stage is the expression of the CD4 and CD8. Okay? There is a positive selection if you already have a functional T cell receptor. Okay. However, it is very important to take note that there's still an MHC restriction. Okay, it's very important. Only cells that react with host MHC will survive. Okay, so we already talk about the major histocompatibility complex. Okay, so CD8 reacts with class 1, CD4 for the MHC class 2. Okay, so they are the only one that survive. Okay, but we have a term called negative selection, and negative selection is equally important because it says here cells that react with self-antigen are destroyed. Why is it necessary to destroy? If we will not be destroying those cells that will react to self-antigen, there would be development of autoimmune disease, and we do not want that to happen. Okay, so cells that react with self-antigen are destroyed. Okay, so the survivors of either positive and negative selection will either express CD4 and CD8. So if they can express now the CD4 and CD8, we consider them as the mature T cells. Okay, CD4 T cells are actually known as T helper cells because T helper cells can react with MHC class 2 and will bind with plasma cells so that they will be able to produce antibodies. CD8 are the cytotoxic T cells. And the function of cytotoxic T cells are actually the same as the natural killer cells because they can kill target cells. Again, let's recall what we mean by target cells. Target cells can be cells that are infected with viruses or intracellular organisms. Target cells can also be tumor or cancer cells. So, cytotoxic T cells can kill target cells by the so-called perforin granzyme system. I think we have already discussed this perforin granzyme system during bacteriology. I hope you can still recall them. And CD8 reacts with MHC class 1. Recall from last week's discussion, MHC class 2 is expressed in antigen presenting cells. MHC class 1 is expressed in all nucleated cells of our body. That is from the last week's discussion. Okay? I hope that's clear. So at this juncture, if you have any question, anything that comes to your mind, please jot it down. Then we'll try to discuss that later. Okay, so let's talk about the T helper cells. Okay, do you know that the T helper cells has the so-called CD4? It is our manifestation that the CD4 can recognize the MHC class 2 proteins. Okay. And it accounts for 2/3 of peripheral cells. Meaning to say, normally, the CD4 CD8 ratio is 2:1. Because we should have more T helper cells than cytotoxic T cells. It accounts for 2/3. Only 1/3 is CD8. Okay. However, in cases of AIDS, it's the terminal stage of HIV infection, the virus targets CD4. Okay? So during AIDS, we have the so-called reverse CD4 and CD8 ratio. So it becomes 0.5:1 or even 0:1. Because in AIDS, the CD4 falls below 200 times 10 to the 9 per liter. And normally, our body should supposed to have between 800 to 1000 times 10 to the 9 per liter. This is the normal CD4 level. But during AIDS, it falls below 200. And remember, if your CD4 level falls below 200, then you'll have what? Opportunistic infections and malignancy, such as Kaposi's sarcoma. Very common in AIDS patients. Okay, so that is your CD4. So there are two types of CD4. Okay, or there are two types of T helper cells. So we have the Th1 and the other one is the Th2. Again, if you will be looking at Th1, Th1 can produce interferon, gamma interferon, and this gamma interferon has antiviral properties, interleukin 2, tumor necrosis factor beta. So the Th1 cells can also activate lymphocytes and macrophages. Okay, so that is Th1. They can activate lymphocytes and macrophages. On the other hand, when you say Th2, they produce a totally different sets of cytokines. So we have interleukins 4, 5, 6, 9, 10, and 13. And incidentally, the Th2 will help B cells produce antibodies because eventually B cells mature as plasma cells wherein they would now become capable of producing antibodies. So at the end of the day, it is the Th2 that can regulate the B cell activity. So Th2 therefore can regulate B cell activity. Okay. Other subtypes of T helpers subpopulations is called T-reg, not T-rexa, T-reg. The word reg actually came from the word regulator. Okay? So, aside from CD4, T-reg would have another cell surface receptor, TCR. We call it CD25. By the way, I've been mentioning CD to you. So CD is actually an acronym which stands for cluster of differentiation. So Treg, aside from CD4, could also have CD25. So why it's called Treg? Treg sometimes is known as the suppressor T cells. So sometimes we call it the suppressor T cells because they suppress the immune response to self-antigen. Very important because if you suppress the immune responses to self-antigen, you prevent the formation of autoimmune disease. And aside from that, Treg will secrete inhibitory cytokines to inhibit proliferation of other T cell populations. Okay, so it can control T cells. Kapag sobrang dami na ng T cells, we do not want it to be, we do not want to have an overproduction of T cells, then it is the Treg that should suppress its proliferation. Okay, so these are the two important functions of T regulatory cells, and we will be able to know if it's a T regulatory cells because of the presence of CD25. Okay, so in summary, this is the an illustration that summarizes our discussion. Okay, so as you can see, um, extracellular pathogens can be controlled by the cells of our immune system. Okay, so let me define here a term, naive CD4. When you say, what do you mean by naive? When you say naive cells, uh, these cells were not able to encounter antigen yet. So since they haven't encountered any antigen yet, so parang inosente pa sila. But once they've seen antigen, they are now activated. Okay? So, and once they are activated, they can participate in humoral immunity and cell-mediated immunity. Okay. Now let's talk about the B cell differentiation. Kanina, we were talking about T cells differentiation. Now let's discuss about the B cells differentiation. Okay, B cells, okay, are produced in bone marrow. T cells are also produced in bone marrow. The difference is that T cells will migrate to thymus to develop. But the development of B cells also occurs in the bone marrow. So unlike T cells, B cells will no longer go out to the thymus. T cells will need to go to the thymus for them to be developed. So it's like the thymus is like the training ground of T cells because this is where T cells will encounter antigen so that they will not become naive. You know why? Because naive cells will eventually die. Okay? So these cells are produced in the bone marrow and its development also occurs in the bone marrow. So differentiation starts with a hematopoietic stem cell. Sounds familiar? This is what you discuss in hematology. And this hematopoietic stem cell develops into an early lymphocyte progenitor. Perhaps you have encountered the term lymphoblast, lymphocytes. So these are the early lymphocytes progenitor. This early lymphocyte progenitor is involved in the development of mature immunocompetent B cells. Take note of the word immunocompetent. Immunocompetent B cells means that they are capable to do its expected functions. And what are the expected functions of B cells? And that is to produce antibodies. However, however, the B cells should be able to recognize antigen. Okay? It has to be activated by an antigen in order for it to become immunocompetent. So if the B cells have not yet encountered any antigen, we call it naive B cells. And eventually, naive B cells will be will actually die. Naive B cells, because you do not have function. Okay, you do not have function, and in the context of immune system, you do not have function, and it's better for you to be eliminated. Okay, so in order for you to be functional, you need to be immunocompetent. And one way of becoming immunocompetent is that you need to be activated by an antigen so that an activated B cells will be differentiated into its functional form. And that functional form would be the plasma cells because it is now capable of producing antibodies. Okay, during the process of B cell differentiation, we have the so-called antigen independent phase, meaning to say, no exposure yet. So when we say antigen independent phase, it means that there's no exposure yet with an antigen. And since there's no exposure yet with an antigen, this can be divided or into, not really divided, but this can be categorized into 4 stages. So we have the pro B cells, the pre B cells, the immature B cells, and eventually it becomes mature. Okay. So as you can see, this is the pro B cells. Okay. So let's discuss them one by one. The pro B cells. The pro B cells develop from rearrangement of genes that codes for the heavy and light chains of antibody molecules. So doon pala nanggaling yung genes, rearrangement of heavy and light chains. So the light chains are other made up of kappa and lambda chains. Okay? So it is made specific for certain antigen by variable region. Okay, so and must successfully rearrange one set of heavy chain genes to become the pre-beta T cells. So in this case, the pro B cells is the stem cell is the hematopoietic cell. Hematopoietic stem cell eventually develops in the pro B cells. During this stage of development, as you can see here in the development of the pro B cells, the pro B cells are still attached to the stromal cell. The stromal cell is actually the cell of the bone marrow, and they are attached to the pro B cell by means of the c-kit. Okay. So in order for the pro B cell to develop into the pre B cell, there should be rearrangement of genes. Okay? There should be rearrangement of C genes. So what kind of genes? Okay? Rearrangement of genes. Okay? Heavy chain and light chains. So the light chains are kappa and lambda. There are not, now that we are an adult, there are actually 5 types of heavy chains. But for this pro B cell chain, pro B cells, it is the mu chain. Okay? So this should be rearranged in order for the pro B cell to become the pre B cells. So must successfully rearrange one set of heavy chain genes to become the pro B cells. And we are referring to the mu chain. okay, or the mu chain. So, in order for the pro-B cell to become pre-B cell, the mu chains has to be rearranged. What is a mu chain? Mu chain is the heavy chain of IgM. Okay? And this particular mu chain accumulates in the cytoplasm and can express on a cell surface with a surrogate light chain. And so the mu chains accumulates in the cytoplasm. So it will be rearranged, and eventually, when this is rearranged, mu chains combine with the surrogate light chain. So parang naging ganito na siya ang itsura niya. So this one is made up of mu and surrogate light chain. Okay? Aside from that, the pre-B cells would also have a short chain of Ig alpha and Ig beta. So, ito yun. So, the pre-B cells, aside from the mu chains in the cytoplasm, it has also some mu and surrogate light chains, and it also has the alpha and beta chain. So, that's actually an indicator that the cell is already the pre-B cells. Okay. So, eventually, the pre-B cell would become an immature B cell. Okay. What's the requirement in order for it to be classified as the immature B cell? Number one requirement, it has to have a complete IgM antibody molecule on the cell surface. Why is it important? Because this is the function. Okay? Antigen receptor. So, kung babalikan natin yung picture, this is now the Ig. This is now the immature B cell. This is now the completely developed IgM. Why did I say that it is completely developed? Unlike here, in pre-B cell, which is just made up of mu and surrogate light chain, in an immature B cell, it is now a completely developed IgM. Okay, so you have the mu chain and you have the kappa and the lambda chain. So this one is very important because this could serve as the receptor for antigen. So this is a receptor for antigen. That's why that is very important. Okay, so it also has a variable regions that determine the specificity for antigen. So we will be discussing in chapter 4 the different types of region or domain of antibodies. But just to give you an idea, okay, so usually this is the FAB, and this is where we can find the variable region. Here, we can find the constant region. Okay, so the variable region here determines the specificity for antigen. Okay, so if you will be looking back, okay, at the illustration, this is the variable region. Okay, that determines the variable region here determines the specificity for the antigen. Okay, so another indicator that the cell is already immature because of the CD21, CD20, and the class 2 MHC molecules. Why? Because B cells are also considered to be as an antigen presenting cells. So you would expect B cells to have MHC class 2 molecules. Okay, so these are the descriptions of the immature B cells. Eventually, the process of central tolerance involves eliminations of B cells that have self-reactive receptors. Okay, why is it again important? This will this will prevent the development of an autoimmune disease. So this will prevent the development of autoimmune disease. Okay? If immature B cells survive central tolerance, they will eventually be moved to the spleen. And spleen will filter blood, and B cells can actually encounter blood-borne pathogens. And if they encounter blood-borne pathogens, that will activate them. Okay? So, again, apoptosis happens for those that react with self-antigen. So, there would be what? Apoptosis, or do not forget the term apoptosis is also known as the programmed cell death. So you now have from pro-B cell, it becomes pre-B cells, then it becomes immature B cells. And eventually, once the immature B cells go to the spleen and develop there, we now have the mature B cells. So the mature B cells develop in the spleen. Okay. So there are two possible fate. One of them is they remain in spleen. If they remain in spleen, we call them marginal B cells. They are important because their spleen filters blood, and marginal B cells can actually quick to react to blood-borne pathogens. Now, another possibility for B cells is that they will go to the lymph node and other secondary lymphoid organs. We call them as the follicular B cells. And eventually, they would also recirculate to these other secondary lymphoid organs. So lymph nodes is an example, tonsils, Peyer's patches, mucus-associated lymphoid tissues. These are all examples of secondary lymphoid organs. So for those B cells that remain in the spleen, we call it the marginal B cells. Okay, so how do we know that the B cells are already mature? So they exhibit two antibodies now. It is the IgM and the IgG. So let's take a look. Okay, this is now the B cells. Okay, so this is the one that remains in spleen. We call it the marginal zone B cells. And the one that enters the circulation to go to the secondary lymphoid organs, we call it the follicular B cells. So how would you know they are mature? They have IgD and IgM in the surface. IgD and IgM in the surface. Okay? So that's how we know that they are already mature. So surface immunoglobulins provide activating signal to B cells when they come into contact with antigen. So contact with antigen occurs, activates B cells. And when they are activated, they are stimulated, and they will now enter the antigen-dependent phase. To give you a summary, antigen-independent phase means, we discussed that a while ago, antigen-independent phase means that no exposure yet. So this happens in pro-B cells, pre-B cells, immature B cells, and at the early onset of the mature B cells. But when the mature B cells encounter antigen, this is now what you call the antigen-dependent phase. An antigen-dependent phase will produce two types of cell, plasma cells and memory cells. So obviously, plasma cells secretes antibodies, while our memory cells are, again, I've been saying it several times, our plasma cells are important in an amnestic response. So they are important in an amnestic response. Okay. So in the peripheral blood, you would really know that they are plasma cells because you'll be able to see um, this cytoplasmic immunoglobulin as in the form of vacuoles. Okay. So plasma cells are able to produce antibodies, and they are found in peripheral and lymphoid organs as well as in the bone marrow. Okay, so to give you again, to summarize, um, surface receptors would also include CD23 for the follicular B cells, CD1 for the marginal zone B cells. Again, marginal zone B cells means that they remain in the spleen. Follicular B cells means that they circulate and went to lymph nodes and other secondary lymphoid organs. Okay, so let us now discuss the roles of T cells in adaptive immune response. The antigen presenting cells are activated during the innate immune response. So it's actually part of our natural immunity. So our T lymphocytes will react with antigen presenting cells so that adaptive immune response will actually occur. And I think I've shown you the animation of that from the last week's lecture. So T cells circulate throughout the bloodstream, lymph node, secondary lymphoid tissues, and they are looking for antigen. They need to look for antigen in order for them to be activated and not become naive, because eventually naive cells will eventually die. Okay? So this is the action of the T helper cells, no? So action of T helper cells, if you will be looking at the illustration, there is one cell that is important in both humoral immunity and cell-mediated immunity. And this is the T helper cell. T helper cell activates cellular immunity, humoral immunity, and also our natural immune response through the process of inflammation. Unfortunately, it is the one being attacked by the HIV. So, if the T cells are attacked by the HIV, both of these types of immunity will be affected, especially if your T helper cells falls below 200 times 10 to the 9th liter. From HIV, we will now enter the clinical stage of AIDS, which stands for acquired immunodeficiency syndrome. Okay, so action of T cells, um, for the cytotoxic T cells, it can destroy the target cells by means of the perforin granzyme system, wherein apoptosis results to the target cell death because of the perforin granzyme system. So that's one action. B cells, on the other hand, respond to T-dependent antigens. Last week, we discussed about T-independent antigens, but T-dependent antigens are those antigens that were presented by the T helper cells by means of MHC class 2. So the MHC, the T helper cells here presented will present the antigen to the B cells. And B cells will eventually become plasma cells to produce antibodies. Okay. They can also respond to T-independent antigen. What do you mean by T-independent antigens? Okay, T-independent antigens. These are T cells that do not need antigen presentation and okay, uh, T-independent antigens are antigens that do not need okay. T-independent antigens are antigens that do not need to undergo T cell presentation and processing. Meaning to say, even without antigen presentation, B cells, through the T-toll-like receptors, can immediately recognize the antigen, even without antigen presentation and processing by the MHC class 2. And the most common example of T-independent antigens are gram-negative bacteria. So majority of the gram-negative bacteria are considered to be as T-independent antigens because they are easily recognized by B cells through the toll-like receptors, even without T cell presentation and processing. Okay, so that is the T-independent antigen. Unlike the T-dependent antigen, you need the help of T helper cell for presentation and processing. But in T-independent antigens, there's no need for that. That's why they are called independent. Okay, now, in the laboratories as a medical technologies, we could be asked to to help diagnose malignancy, particularly for, I hope you still remember your acute lymphocytic leukemia, L1, L2, L3, and even the chronic lymphocytic leukemia, or even lymphoma, immunodeficiency disorder, and AIDS. In AIDS, doctors would really want to know your CD4, CD8 ratio, and also the amount of the lymphocytes. And in order for us to identify lymphocytes, we cannot just look at the peripheral blood in the blood smear. We need a special type of machine known as the flow cytometry because it can segregate lymphocytes into subset by means of monoclonal antibodies specific to a certain type of lymphocytes. Okay? So there you have it, our discussion in the T cell differentiation and B cell differentiation.